Good morning, dear students. Today we are going to study about control of microorganisms using physical agents. Control of microorganism by physical agents aims at using various physical methods used to control the growth of unwanted microorganisms. Control of microorganisms is essential to prevent the transmission of disease, spoilage, and contamination. So, control of microorganism using physical agents aims at using various physical methods like temperature, pressure, radiation, filtration, etc. to control the growth of harmful microorganisms which spread diseases or which cause spoilage of food materials, which result in contamination and decomposition. Various physical agents that are used to control microorganisms include temperature, pressure, desiccation, radiation, filtration, etc. First one is temperature. It is a very common and effective way of controlling microorganisms. Microorganisms have a minimum, optimum and maximum temperature for their growth. Very low temperature inhibit microbial growth by slowing metabolism but do not kill them. High temperature denature microbial enzymes and proteins. So optimum temperature is ideal for the growth of microorganism. Both high temperature and low temperature, it inhibits the growth of microorganism. In very low temperature, the enzyme activities get slowed down, the metabolism gets slowed down, but it doesn't kill the microorganism. But high temperature, it will denature the microbial proteins and enzyme and thereby killing them. So temperature can be used as a very effective method for controlling the growth of microorganisms. Thermal death time is the shortest time required to kill all the test microbes at a specified temperature. Thermal death time is denoted by TDT and it is the shortest time or the minimum time that is required to kill all the microorganisms that we have taken at a specified temperature. And thermal death point, it is the lowest temperature required to kill all microbes in a sample in 10 minutes. So, in thermal death point, we are specifying the time as 10 minutes and we are applying a lowest temperature that is required to kill that microorganism within that 10 minutes of time. And various methods that are employed based on the temperature include heat, pasteurization, autoclave, boiling, tyndallization and freezing. Heat. Two types of heat are used for sterilization or killing of microbes. It includes moist heat and dry heat. Moist heat kills the microorganism by coagulating their enzymes and protein, and it also melts the lipid content in the cytoplasmic membrane. It has a lower temperature and shorter exposure time. Water vapor present in the moist heat facilitates the penetration of heat into materials and it is more effective in killing than dry heat. So, in moist heat, it kills the microorganism by coagulating the proteins and enzymes present in the protein and also it helps to melt the lipid that is present in the cytoplasmic membrane of the microorganism. And this moist heat has an advantage that we are applying moist heat in low temperature and the exposure time is also very shorter. And in moist heat, kill the microorganism by having a water vapor present in the moist heat. It helps in the penetration of this heat into the microorganism. So the penetration of this heat into the microbial cell make it more effective in killing than applying dry heat. So the peculiarity of moist heat is that we can apply moist heat with very low temperature and the exposure time is also very short, but the penetration into the microbial cell is very high when compared to that of dry heat. Then dry heat. Dry heat kill microorganisms through a process of protein oxidation. Dry heat uses higher temperature than moist heat. Example of dry heat application is in incineration using electric heating coil. Then in dry ovens also we are applying this dry heat and here the temperature should be around 150 to 180 degrees Celsius. Dry heat sterilization are used as aseptic techniques in laboratory. So dry heat, it is used as a sterilization technique in laboratory. It is applied in dry ovens as well as incinerators, and this dry heat, we are applying a higher temperature than the moist heat, and temperature around 150 to 180 degree is needed, and the exposure time is also very high, but the penetration is low, and here the microorganisms get killed by a process of protein oxidation. Next is autoclaving. It is more effective method of moist heat sterilization technique. It works on the principle of steam under pressure. Here steam directly come in contact with the material to be sterilized. Autoclave is sterilization technique where materials are placed under 15 pounds of pressure, and the boiling point of the water is raised to 121 degrees Celsius for 15 to 45 minutes, and autoclaving kill both vegetative organisms as well as endospores. So autoclaving, it is used as a sterilization technique in all the microbial laboratories, and it works on the principle of steam under pressure, and it is an effective method of applying moist heat for sterilization technique. And here the materials to be sterilized come in contact with the steam that is generated, and steam is generated by boiling water above its boiling point, that is up to 121 degrees Celsius. Water is being boiled for about 15 to 45 minutes, and at that time the pressure generated will be 15 lbs, that is 15 pounds of pressure will be there, and that within that high temperature and pressure, most of the vegetative as well as spores get killed. So it is one of the most important sterilization technique that is used to sterilize all the contaminated microbial petri dishes after experiment, as well as sterilization of the media before culturing in microbial laboratories. Next one is boiling. Boiling means we are boiling the water up to 100 degrees Celsius. It will kill all the vegetative cells within 10 minutes. But some viruses and some other microorganisms can survive the boiling temperature, especially the thermophils wouldn't get killed at this high temperature. So hepatitis virus, then spores of the Clostridium and Bacillus, they may survive this high boiling temperature. Hepatitis virus can survive boiling water up to 30 minutes, and Clostridium and Bacillus, they can survive the boiling temperature for even hours. So boiling would not remove Clostridium and Bacillus species, so it can survive boiling temperature for hours. So temperature above 100 degrees Celsius is required for killing this hepatitis virus, Clostridium and Bacillus, some Bacillus species. Pasteurization, it is a mild heating of milk and other materials to kill particular spoilage organism or pathogens. We have already studied various pasteurization techniques, and here we have dealt only with two types of pasteurization, and it doesn't kill thermophiles. Pasteurization is not a complete sterilization procedure. It only avoids some of the mesophilic microorganism, but it doesn't kill the thermophilic microorganism, because we are just heating milk or other fruit juices all to a temperature up to 72 degrees Celsius. So most of the thermophilic microorganisms will survive at that temperature. So we are, after pasteurization, we are immediately freezing that drinks in a cold storage area, so as to avoid the growth of these thermophilic microorganisms. So in pasteurization, we are either using flash method or low temperature holding method. Mostly ultra pasteurization is also there, and in flash method, that is high temperature short time method, we are heating the milk up to 72 or 71.6 degrees Celsius for 15 seconds, and in low temperature holding method, we are heating the milk to 62.9 degrees Celsius for 30 minutes. Next is fractional sterilization or tyndallization. Tindelization, here we are using sterilization intermittently. That means we are repeatedly sterilizing substance that cannot withstand that autoclaving temperature. So the materials that has to be sterilized is subjected to a free flowing steam for 30 to 60 minutes, and then it is after 30 to 60 minutes of steam exposure, we are incubating that material for 23 to 24 hours and then subjected to steam again, and repeat the cycle for 3 days, and it is used for preserving canned foods as well as laboratory media. So tindelization, it is otherwise known as fractional sterilization. It is called fractional sterilization because we are not applying the temperature directly into a material continuously. We are applying temperature repeatedly for 3 days with the incubation period of about 24 days. So I mean 24 hours, so that the materials wouldn't get damaged by the application of extreme heat on them. So in this tindelization procedure, the materials are exposed to steam for about 30 to 60 minutes, and after that they are kept aside or they are incubated for 24 hours, and then they are subjected to steam again for 30 to 60 minutes in the second day, and then it is again incubated for 24 hours, and after in the third day also it is been exposed to the steam for 30 to 60 minutes. So we are intermittently applying this steam for 3 days, so that it is called a fractional sterilization, and this method is used to preserve canned food items as well as laboratory media. Next is freezing or low temperature. So low temperature also inhibit the growth of microorganisms by slowing down the microbial metabolism by deactivating the microbial enzymes. So refrigeration at 5 degrees Celsius slow down the growth of microorganisms and keep the food fresh for a few days. Freezing at minus 10 degrees Celsius stop the microbial growth but generally does not kill microorganisms. So even at minus 10 degrees Celsius, microorganism wouldn't get killed. That means in our refrigerator also there exists microorganism that will be in a frozen state, that is they are only in an inactive state. Still they are active when the temperature will be back to the normal room temperature. Keep food fresh for several months. So freezing can keep the food fresh for several months. Laboratory and medical specimens may be frozen on dry ice or at ultra low temperature for storage and transport. So we are using this freezing or low temperature method for keeping laboratory as well as medical specimens for storage and transport, and for that purpose we are applying dry ice. Dry ice means it is a solid form of carbon dioxide, and here solid carbon dioxide is used as a preservative, and it gets frozen very rapidly than water ice, and or we are using ultra low temperature, that is temperature below minus 16 degrees Celsius for storage and transport. Next is pressure. Exposure to high pressure kills many microbes. In food industry, high pressure processing is used to kill bacteria, yeast, mold, parasite and other viruses in food. It helps in maintaining food quality and extending the shelf life, and application of 800 MPa is sufficient to kill vegetative cells by protein denaturation. Endospores may survive these pressures. We are applying high pressures to kill many microorganisms like yeast, mold, parasites, virus, etc. present in the food, and application of pressure to kill these microbes is called Pascalization. So Pascalization is the application of high pressure to kill microbes or microorganisms, and it helps to increase the keeping quality of the food, and it extends the shelf life of the food. And in Pascalization we are generally applying 800 megapascal it is 4 megapascal of pressure to kill the vegetative cells by protein denaturation. So by in pascalization technique, the microorganism get killed by by their protein denaturation. But endospores can survive in this pressure. So it kills only the vegetative cells. Endospores will still survive in this pascalization pressure. Today we have dealt with various types of physical agents that are used to control microorganisms. First one, we have dealt with the temperature. The temperature itself, we have gone for different variations of temperature like moist heat, dry heat, then pasteurization, autoclave, boiling, then tyndallization, freezing, etc. And we have also seen the pressure as a physical agent to control the microorganism by pascalization method to kill various microorganisms. It helps in maintaining the food quality. And here microorganisms are killed by protein denaturation, but it is not effective to kill endospores. So that is all for today's class. We will continue the control of microorganism by using physical agents in next class. Thank you.